Okay, so I would like to welcome you all to the fourth webinar of the Israeli Translational Research Network. Thank you for joining us. I'm Ruth Peretz. I'm a medical oncologist and physician researcher working in Rambam Medical Center, focusing on gynecological malignancies. And I'm the member and I represent here the Clinical Research Institute in Rambam, or CRIL for short, a research institute that gathers most of the physicians researcher in Rambam that perform basic and translational research. This is the fourth webinar of the Israeli Translational Research Network, and it will be recorded and uploaded to the Israeli Translational Research Network website. Please feel free to ask questions using the Q&A feature at the bottom of your screen, and Professor De Souza will answer all of your questions at the end of his talk. So I'm honored to present today our speaker, Professor Martin De Souza. Professor De Souza graduated with a BS in pharmacy from the University of Bombay, India, and then completed his Ph.D. from the University of Pittsburgh, Pennsylvania. He then joined Mercer University, Atlanta, and is currently a professor and director of graduate programs. He is also the Endowed Chair of Pharmaceutics and director of the Mercer Clinical Laboratory and co-director of the Center for Drug Delivery in Mercer University in Atlanta. Dr. De Souza's Nanotechnology Laboratory mainly focuses on the design and delivery of both oral and microneedle-based transdermal vaccines for infectious diseases and for cancer. Currently, Dr. De Souza's lab is focusing on vaccines for infectious diseases such as SARS-CoV-2, influenza, RSV, Zika, gonorrhea, and just to name a few. Additionally, work is underway on therapeutic vaccines for cancers such as melanoma, ovarian cancer, prostate, and breast. Dr. De Souza has already graduated around 60 PhD students and has published over 120 manuscripts. He has been the recipient of several research grants from the National Institute of Health, the American Diabetes Association, the Georgia Cancer Coalition, the Georgia Research Alliance, and he served as an editor and on the editorial boards and the junior reviewer for over 10 scientific journals. He has several patents issued in the area of nanotechnology. Currently, he has an active NIH-funded R01 and SSTR research grant. His current work on the COVID-19 research involves designing two novel vaccine formulations using the spike S1 surface glycoprotein and hit inactivating SARS-CoV-2 virus. Thank you, Professor De Souza, for coming to speak with us today. And I'm giving you the floor.
Wonderful. Good afternoon, all of you. It's morning for us here in the US Eastern Time. But I want to begin by thanking Dr. Yehudai, Reshef, and Dr. Peretz for inviting me for this presentation. I will begin my sharing my screen now. Let's see. Okay. Just want to make sure that, yeah, let me go up. All right, so just want to make sure that all of you can see the screen and can hear me clearly. So one of the things that my lab basically specializes in is designing vaccines, mainly particulate vaccines. And over the years, I've sort of worked on vaccines for cancer. For about 20 years, I did almost exclusive design of cancer vaccines. And we do have one ovarian cancer vaccine that will begin its phase 1 clinical trial sometime later this year. And that'll be the first of its kind ovarian cancer vaccine that will be entering the clinical trial phase. But in addition to that, I've more recently worked a lot with vaccines for infectious diseases, including influenza, RSV, gonorrhea, and more recently, of course, since March of last year, on the COVID-19 vaccine. One of the things that we are doing is, and as most of you are familiar, all these vaccines that are available for COVID-19 are essentially very difficult to, to store. They all need cold storages of almost unreal magnitude. it's what we have at the present time. So my lab is basically focused and trying to, move more towards designing vaccines that are very stable for long periods of time. They don't need cold storage. And more importantly, we are also working on vaccines using essentially, let me get this laser pointer here. So essentially using single microneedle based vaccine. And these vaccines are applied more like a Band-Aid patch, if you will. And we'll talk more about that in just a moment. And one of the more recent ones, we are also designing oral dissolving films. So ODF, oral dissolving films, again with the vaccine microparticles. Big advantage there as well. We can, they don't need cold storage because the vaccine is in a dry form. So let's take a look and see what these vaccines are made up of. So all these nano vaccines basically are composed of a biodegradable, biocompatible matrix. And this can be of several types. It can be albumin, beta-cyclodextrins, polyvinyl alcohol, polylactic glycolic acid, trehalose, sugars, all kinds of different stuff. Anything that's biodegradable, that's critical. We can then incorporate the vaccine antigen, whether it's a whole virus, whether we are looking at specific proteins that we've identified. And within these nano vaccines, we can also add adjuvants such as IL-2, any cytokines. We can also add, the regular alum, all of those typical adjuvants that we have. And once we formulate those, the advantage, as I said, is it's a dry powder form, no cold storage is needed. And because it's particulate in nature, we essentially have slow and sustained release of the antigen. Now, because they are particulate in nature, of course, they are highly immunogenic because they are not in a soluble form. They can't be taken up into the, by any mechanism other than antigen presenting cells. So in other words, macrophages and dendritic cells play a major role in their uptake. And so the delivery is very, very strong. We make these particles using a spray dry, and I'll talk more about it. And as I said, we're using a Band-Aid patch, much like the one you see in this picture out here. And this can be self-administered. So that's a big plus as we talk about vaccines, in the long run. And as we can see, some of these vaccines and some of these infectious diseases are going to be here for a long, long time. When you look at the differences between these vaccines, if you look at a typical subcutaneous or intramuscular vaccine, this is what a hypodermic needle looks like on a comparative scale. And you notice that it goes right down into, the tissue. These are where the blood vessel and the nerve endings are. And so you experience a lot of pain. On the other hand, if you look at the microneedles, they barely make it into the dermis. They cross the stratum corneum, which is the top layer of the skin. They get into the epidermis and dermis. And here we see a lot of the antigen presenting cells on the skin. There are a large number of what we refer to as Langerhans cells, which are very specialized skin immune cells that are present in very large numbers. And the goal here is that we administer these microneedles, and then the microparticles get out into this region and are taken up by the Langerhans cells, which we know then drain into the lymphatic system, the spleen, so on and so forth, to essentially exert their effect. So when it comes to COVID-19, as I said, in March, the pandemic was declared, and so began basically a series of studies to see what best could be done to design a vaccine. So one of the early things that was discovered was this SARS-CoV-2 has a very unique S1 protein. It has S1 and S2 protein that are sitting on the surface. And by way of the mechanism of infection, this S1 protein attaches to ACE2 receptors, which are present in several cell surfaces, and we'll see some of those. So if you look at these cell surfaces, I mean, the human body is designed really as the perfect target for these, for these SARS-CoV-2 viruses. If you look at it, we have ACE2 receptor expression in the CNS, in the airways, vasculature, in the lungs, liver, gut, kidneys, heart, eyes. There's really no organ that is spared. So it's like almost a natural target for the human system. And so it can infect very, very easily. And that's why we have all these huge number of infections which are difficult to control at the present time. On a similar basis, we also have a large number of vaccines in different stages of clinical trial. And so if you look out here, there are, viruses, vaccines made up of entire viruses, nucleic acid. We see mRNA, DNA vaccines of the kind that are on the market right now. There are several protein-based vaccines, a lot of them in clinical trials, and viral vectors. So, people are using all kinds of different approaches to basically try to tackle this issue. What we've done in our lab is we basically are looking at the spike S1 protein or the heat inactivated SARS-CoV-2 virus. And the reasons are spike S1, as we said, is the major attachment protein. If we can design a vaccine for that, then we can block that part. And that looks like a pretty straightforward approach. And the second one is heat inactivated. We're using the entire virus structure because then we are not excluding any of the proteins that may be excluded if we just used one of these. So we are looking at both approaches. And again, the overall goal is to make a Band-Aid patch so that it's in a very stable form. In terms of overall study design, we are going to formulate the vaccine, characterize it, do some in vitro testing, and then finally test them in vivo. By way of manufacture, we make the microparticles or nanoparticles using a Buki spray dryer. Again, this is a completely automated process, so we don't have any day-to-day variations. The vaccine solution is put into this flask and we can have a continuous process. And it's sprayed through this chamber here. It collects in the cyclone separator and this container, and we can add larger containers as well. And the nice thing is this entire assembly can be kept into a laminar flow hood or a sterile area. And so you can make sterile batches rather easily, again, completely reproducible. And as I said, the final product is dry microparticles. So all the liquid is evaporated. And so it's then very stable beyond that point. Once we make the microcaps particles, we basically look to characterize several parameters like size, shape, uniformity. We look at the charge, the encapsulation efficiency, stability, and release studies. In terms of the overall yield, we got excellent yields, higher than 95%. Likewise, the encapsulation efficiency was in the 90s. And the size was less than 1 micron. And again, we can make and design sizes of any kind based on the polymer and the concentration. And here are some SEM images of the spike S1 vaccine antigen particles. And as you can see, they're pretty uniform as they are formulated. One of the things that we were interested in looking at the half-life of release of the S1 protein from these particles. And what we noticed was the half-life of release is about 50 hours or approximately 2 days. So that's good. We want a slow release of the particle, but we don't want it to be so slow that it comes out over many days or weeks. So 24 hours, 2 days is excellent timing. it gives enough time for the particles to be taken up into the phagocytic cells and the immune cells of the body. One of the early studies was to, of course, look and make sure that they were non-toxic. And here you can see there's no toxicity at all, even at very high doses. Pretty safe to use. We basically look at, try to study these microparticles in vitro by looking essentially at characterizing what happens when the particle is exposed to dendritic cells. If the vaccine particle is potent and displaying an immune response, we get a release of nitric oxide, which we can measure. We are also interested to see if these vaccine particles in vitro can basically generate Th1 pathway or Th2 pathway. And again, depending on the type, Th1, we are looking at MHC class 1 or CD80-86 response. And then the cytotoxic CD8 positive T cells will increase. On the other hand, if the vaccine generates an MHC class 2 pathway, then we'll get helper CD4 positive T cells that will be induced. And this is very important. So if a vaccine generates both of these, that's even better. But we need to characterize this in vitro because we really don't want to waste too many animals. So this is a system we've developed in-house to nicely characterize this. And here you can see the nitrite release. And if you look at the marketed, we used a control, which is the marketed measles vaccine. And here in the last column, you can see when the virus particles in the presence of alum and MF59 as adjuvants, you get literally a double response here of the nitrite release. So in other words, we are getting a strong immune response. We also looked at the MHC, again, class 1 and CD8 response. This is the co-stimulatory molecule related to MHC class 1. And again, we can see in this last column here, for both CD8 and MHC1, the response is very high in terms of when the virus and the alum, which is the adjuvant, and MF59 are used. And again, in all cases, these are much stronger than the equivalent doses of the measles vaccine, which we use as our control. Much lower responses were seen when the virus suspension was used without microencapsulation or without particles. A similar response was seen in the case of the MHC class 2 and CD40. So in other words, what the take-home message here is that we get a very strong Th1 and Th2 response when we use these microparticles for these viruses and spike 1 proteins. We further confirmed this by looking at autophagic vacuoles, and we saw that the SARS-CoV-2 virus vaccine particles induce strong autophagic vacuoles. Okay, and you can see these bright green particles, and these are good because if it induces autophagy, that means it's producing a good immune response. Next, we make the vaccine basically the microneedle patch. And to do that, we first create a microstructure using the microneedles, stainless steel microneedles. And once we make all of this, we then use a 3D bioprinter basically to use that to convert it, I would say, to a semi-automatic process. So we have we try to minimum the batch-to-batch variation by using a bioprinter for this process. And again, we used dissolvable microneedles. We can use any polymers. There are several to choose from, including PVA, mannitol, trehalose. Again, these are sugars. We want these to dissolve rapidly. We then move on to characterize the microparticles. And again, this is an SEM of the individual microparticle. And you can see they are very well formed. And when we put them on the skin and stain it using methylene blue dye, you can see very clearly. We can also do confocal microscopy and look at the depth of it. And it's about 600 micrometers in depth, which is, just perfect for what we need. Then we also looked at the entire patch. This is an SEM of the entire patch. And what we found that after application of this microneedle patch to the skin, they release the entire needle is dissolves in about 2 minutes or so, which is good. We really want quick release. From there on, we looked at the microscopy of the skin part, and we can see this indentation here where the microneedle goes in, and it goes past the stratum corneum and the epidermis into this region, delivering the payload out there. One of the nice things is when we put the microparticles in the microneedles and then did the release study, we found that the release of the half-life was around 2 days. In other words, the microneedle was not slowing down the release from the microneedle, from the microparticle, which is good. We don't want the microneedle to slow down the release, but it should be comparable to the microparticle. And then finally, we also went on to do look at some animal studies, and the take-home message here is we looked at the serum IgG at week 4 and 12, and for both the S1 microparticles in the microneedles as well as the virus in the microparticles, we did see a good immune response, and those are much higher than when the virus was just added in its suspension form. Okay, in both these cases. So in the microneedle, we got the highest response, which is really good. This is what we were looking for. With that, that's where we are with the COVID-19 project right now. And we are, continuing to look at some studies in larger animals and basically using, prototypes, basically to create a more marketable formulation. Some other studies we've done over the years are looking at an RSV vaccines using VLP, and that my lab has been working on that. We're also looking at this was a universal flu vaccine that we were trying to design, an RSV vaccine, and as I mentioned before, the ovarian cancer vaccine. None of this work, of course, could be done by me alone. In fact, I have huge numbers of graduate students over the years. And currently I have about 10 PhD students and a lab manager who pretty much handle most of the work. With that, I'll be glad to take any questions. Feel free to type your questions in the chat box so we can basically look at that. Again, I want to thank you for your time and attention. So yeah, go ahead if you have any questions.
I'm just not sure if the Q&A feature works. So maybe I'll just start as a ovarian cancer researcher and a phase 1 researcher. I would be very happy to hear what you can tell us about your coming phase 1 clinical trial in ovarian cancer.
Yeah, we are pretty excited about that. We are hoping to get some good results, and we are those studies are will start at the MD and Anderson Cancer Center in Houston. And we have about 8 oncologists on board. So we're excited about that.
But I don't want to get into too many secrets, but what is the epitope you're injecting?
Oh, so for those, we have identified about 6 different antigens that so what these oncologists have done is in ovarian cancer patients, they've identified about 5 to 6 antigens that are common in all of these patients. And once they've identified those, they have then gone on to synthesize those antigens, in the lab, and those will be used as the vaccine prototype in the phase 1 clinical trial. So yeah, and we're trying to do our best to include as many as we can in that phase 1. I see some questions here in this box. So it says, did you try, so one of the questions is, did you try to use peptides instead of the entire protein? Yeah, so one of the things that we're also doing, as I said earlier, is in addition to the S1 protein, spike protein, we are also using the entire virus. One of the advantages there is, and we've seen that in many other studies before with the influenza virus as well, is you can catch all the epitopes in actually using the entire virus. So yeah, we are also looking at other peptides and synthetic peptides that we are collaborating with other people as well. And it's the next question is, can we use the anti-SARS-CoV vaccine concept to inhibit the immune response to patients with severe COVID-19? So let's see, anti-SARS-CoV vaccine concept. I guess in a way you can. So, I guess one of the issues that we are faced with here is there is this over response of cytokines, the classic cytokine storm that we are seeing. And that's something that I guess we are going to have to learn to deal with. And one of the things that we are also working on is to use an antisense oligonucleotides to a bunch of cytokines like TNF, IL-1 beta, IL-6. And what we found was we can actually basically blunt the cytokine storm. And that will be an interesting thing to look at in conjunction with basically treating these patient profiles. I think many patients are literally dying of toxicity due to the cytokine storm. And that's something I think none of the current vaccines are able to address, unfortunately. But I'm sure there are people working on that, and we should sometime later this year be seeing several approaches along those lines. Good question.
Thank you very much. Any more questions from our audience? Okay, so if not, thank you very much, Professor de Souza, for a fascinating science fiction-like talk.
Thank you. I wish I had more time.
It was really, really amazing. And I hope I'll have the chance to participate in one of your clinical trials at some time. We have a wonderful phase 1 unit in Rambam Medical Center.
That's wonderful. I'll keep that in mind.
And Neta, can you put on the next slide? Do we have Neta?
So I may go ahead and leave the meeting if that's okay.
Yes,
I have a class to teach. Yeah. Thank you so much again.
Thank you very much. I just wanted to remind everyone that the talk will be uploaded to the website of the Israeli Translational Research Network, so everybody will be able to see it at their own time. Thank you very much again for speaking with us today.
Thank you.
Bye-bye.